Right. Good evening. Good evening, people. Can you hear me clearly? Can you hear me, people? Do you hear me? Good evening. Good evening. Komada. Yeah, I'll share the PDFs. No problem. Um, yes, you can hear me. Right, people. We were discussing. We have to discuss. We have to discuss paper 5. The paper 5. You answered the paper. I think your papers are marked. Please check in the folder. Can you give me a feedback? I couldn't check today. Uh, have you received the marked paper? Did you check in PDF? Sorry, did you check in the personal folders? Yes or no? Personal folders, marked paper. Amira says receive the marked paper. Right. So we'll do it on this way. Right. You are answering and then submitting. We are marking and then making it available in the personal folder, and then we discuss. It looks like okay, ne? Right? It looks perfect, ne? So you, after marking and everything, people, we can discuss your errors. I know that you get less marks because there are certain things that you have to fulfill. You have to study, and then you have to practice these question answering and all. So don't let yourself down. Just go on doing this with some interest, so then you will improve your marks, right? When time to come, you will improve your marks. So let's continue, people. Today we have to discuss this paper. This is the marking scheme. So where is the paper? Don't expect that you're going to like improve your marks, people, overnight. Since it's a slow process, it takes time. That is why you have to like practice it. If you can do it overnight, there is nothing to practice. You can straight away go on answering. But it will take some time to improve, right? But I can guarantee your improvement if you put enough attention, interest in this work. All right, dear. Sir, is that a flight engine sound? Is there any noise come? Any noise come? My voice is clear, ne? Clear, right? Toffee bro, what's that? TB. Is there any background noise comes? I will continue with the discussion, people. First question. First question, select the correct statement about carbohydrates. All are reducing sugars. All are reducing sugars. Correct statement, wrong. And then all are having glycosidic bonds. No, wrong. And then the ratio between hydrogen to oxygen ratio in carbohydrates 2:1, that is correct. The simplest carbohydrate has 2 carbon atoms, no, 3, neither. We said 3. And then people, all carbohydrates are macromolecules. All carbohydrates are macromolecules. What do you say about that? Wrong, neither. Only polysaccharides are macromolecules. So we got the answer now. Third one is the answer. Question number 2, which of the following polymer act as a storage material in fungi? Storage material in fungi, people, they have mentioned as glycogen, so we will take the answer as the fourth one. Question number 3, proteins. Proteins form secondary structure due to disulfide bonds. So your voice is going. Venuk. Check one, check one, check one. Check one, check one. Check one, check one. No, I can't fix it, people. I cannot fix it. We'll continue like this, huh? I'll have to go like that. Check one. Check one. That's nice, neither. Echoing. That's nice. So we'll go with that. Right. Next question. Next question. Question number 3. Proteins form secondary structure due to disulfide bonds. Secondary structure is not due to disulfide bonds. You know, hydrogen atoms, hydrogen bonds are formed between the distant oxygen and hydrogen. Hydrogen attached to nitrogen, and between the oxygen of the carbonyl part. So, secondary structure due to disulfide bonds wrong. Secondary structure is due to hydrogen bonds between the distant amino acid. They are made up of 26 wrong, 20 different amino acids are there. Carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus is wrong and would not be denatured by detergents. Would not be denatured by detergents. No, they are denatured by detergents. Wrong. Contribute to transport of materials, correct. So I'll take fifth one as the answer. For question number 3, fifth one is the answer. Question number 4, which of the following statement regarding lipid is correct? Lipids are macromolecules containing carbon, hydrogen and oxygen. They are macromolecules containing carbon, hydrogen, oxygen. Lipids are large molecules, but you can't take them as macromolecules, wrong statement. Each hydrocarbon of saturated fat contain one double bond. Each hydrocarbon chain contain one double bond, wrong. There can be several double bonds. And then when fat are formed, glycerol and fatty acids are joined by hydrogen bonds. Glycerol fatty acids are not joined by hydrogen bonds, wrong. Hydrogen to oxygen ratio of lipid is higher than 2:1. That is correct, people. More hydrogen present than the oxygen. Correct statement. And 2 phosphate groups are present in phospholipid molecules. 2 phosphate groups are present in phospholipid molecule, wrong. 1 phosphate group is there. So we got the answer now, fourth one for the fourth question. Question number 5, ice floats on water because molecules on ice are further apart than the molecules in liquid water, that is correct. People, there is a expansion of volume of ice upon freezing. So we learned that this is not happened with the other, but it is like some unusual feature that we can see in water. Volume of water, and if you compare the volume of ice, you can see that ice has greater volume because of this the formation of the crystalline lattice. Then the distance between the water molecules are going to increase. Water surface tension is strong enough to support ice. No, that is not the reason. And then the oxygen, there is an oxygen, what is that? Indus all. There is an oxygen trapped. I don't know this word. Indus all in the liquid water. I think entrapped or something near the oxygen. Uh, all the liquid. There is a spelling mistake here. Anyway, wrong statement. Liquid water is less denser than ice. No, liquid water is more denser. Wrong. Liquid water has minimum density. No. Maximum density at 4 degrees Celsius. Wrong. So we got the answer now. First one is the answer for that question. Moving to question number 6. Some nucleotides contain hexo sugars. Some nucleotides contain hexo sugars. No, we said pentose sugar all the time. Act as organic cofactors. Act as organic cofactors. Act as organic cofactors. Nucleotides NAD, FAD, NADP. Possible. Act as organic cofactors. Possible. Serve as an enzyme. No. Nucleotides can't work, but some RNA can work like enzymes. As act as oxygen carriers. Nucleotides not oxygen either. Hydrogen and electron carriers. Wrong. Serve as food reserves. No. So we got the answer, people. Second is the better answer. Question number 6 answer is second. Moving to next set of questions. On the other page, contain only 2 branch polysaccharides. Contains only 2 branch polysaccharides. Sorry, contain only branch polysaccharides. Branch 1, glycogen is branch, cellulose is unbranched. Cellulose is unbranched. Hemicellulose, amylopectin, hemicellulose is branched, amylopectin is branched, glycogen is branched. So I think we got the answer now. Third one, amylose, glycogen, cellulose is unbranched. Cellulose is unbranched. So you can take seventh question, third one as the answer. Question number 8, the compound which does not contain nitrogen as the constituent element is OR. Compound which does not contain nitrogen as a constituent element. Pectin, no nitrogen. mRNA, nitrogen present. Casein, nitrogen present. Does not contain neither. And then chitin, nitrogen present. Actin, protein, nitrogen present. So we got the answer now. First one for question number 8. Question number 9, characteristic features of green algae is. Characteristic features of green algae. Sporophyte produce spores that contain a wall. Sporophytes produce spores that contain a wall. Sporophytes produce spores that contain a wall. Characteristic feature of green algae. Wrong, neither. Multicellular gametangia. No, algae they have unicellular gametangia. Dependent embryo. No, plant has that. Cell wall present. Apical meristem, no apical meristem. So the answer is going to be fourth one. Question number 10. 4 structures present in protist are as follows. Multicellular thallus. Multicellular thallus. Multicellular thallus. Organisms having A, B, C, D are respectively. Multicellular thallus, contractile vacuole. Contractile vacuole, you have to take people, euglena, amoeba, paramecium. Pellicle present in euglena and pellicle present in paramecium. Cell wall, sargassum, ulva, multicellular thallus, and then contractile vacuole, diatom. First one we can reject. Diatoms don't have contractile vacuole. Euglena, contractile vacuole, paramecium, pellicle, and gelidium cell wall looks like okay. Gelidium, amoeba, ulva as pellicle wrong. Sargassum, multicellular thallus, paramecium, amoeba, pellicle wrong. Ulva, euglena, sargassum, pellicle wrong. So we got the answer. 10th question, second is the answer. Question number 11, which answer include respective order of protist bearing following characteristics? Which of the following answer include the respective order of protist bearing following characteristics? Having contractile vacuole but no food vacuole. Contractile vacuole but no food vacuole. Paramecium has food vacuoles. Euglena, contractile vacuole, no food vacuole. Bare chloroplast, euglena. Golden brown in color, sargassum. Wrong, diatom. Having contractile vacuole but no food vacuole. Paramecium having contractile vacuole, no food vacuole. No, we can't take first one. Euglena having contractile vacuole but no food vacuole. Amoeba having contractile, you can't take amoeba. Euglena having contractile vacuole, no food vacuole. Bare chloroplast, ulva, okay. Because it has meganucleus and micronucleus, got to be people paramecium. So paramecium at third. Paramecium at fourth. Paramecium got to be at fourth. And then golden diatom has to be third. Diatom has to be third. Diatom is third. I think fourth one for question number 11. Do you agree with that? Right. So question number 11. Euglena, it repeats, having contractile vacuole but no food vacuole. Then bare chloroplast and then golden brown, sargassum wrong. Paramecium, you can't take at the front, the contractile vacuole but no food vacuole, we can't take that. So we got the answer, fourth one here. Question number 12. Which of the following correct regarding organisms, specific features and habitat? Correct regarding organism, specific feature and habitat. Diatom cell walls with silica. Marine and freshwater. Which of the following is correct regarding organisms, specific features and habitat? Diatoms cell wall with silica live in marine and freshwater, correct. Ulva thallus is greenish red color, wrong. Paramecium presence of one or two flagella, wrong. That's euglena. Euglena eye spots live in terrestrial freshwater marine, wrong. Sargassum gasfill floats live in freshwater, wrong. So we got the answer, first one. Question number 13. Which of the following is found in both DNA and tRNA? Double helix structure, no. Ribose, no. Sugar phosphate backbone, yes. Complementary base pairing, no. But that's present in tRNA. And thymine, no. So what is your answer for question number 13? I think best answer is the third one. Best answer is third. If you have any arguments, please send them. True statements about the compound contain the organism is are. True statement about the organism contain in organism is a statement about compound containing organisms. Some storage polysaccharides are formed by polymerization of monosaccharide belongs to aldose group. Some storage polysaccharide. Now we can take starch formed by polymerization of monosaccharide belongs to aldose. Glucose is aldose, correct me? Glucose is aldose, correct. Respiratory pigment of all vertebrates are proteins, and they have quaternary structure. Respiratory pigments of all vertebrates are proteins that have quaternary structure. Myoglobin has tertiary structure. I think you haven't learned it yet. So all respiratory pigments of all vertebrates have quaternary structure wrong. Can you write there? Myoglobin is having tertiary structure. And then people, oxygen atoms and hydrogen attached to nitrogen atoms contribute for formation of hydrogen bonds. Oxygen atom and hydrogen attached to nitrogen atom contribute for the formation of hydrogen bonds in the secondary structure of the protein, correct. The spider silk fibers are formed by twisting and bending of polypeptide chains, correct. The spider silk fibers are formed by twisting and bending of the polypeptide chain, wrong neither. Reason is this is secondary structure. Secondary structure. So I'll take it as wrong. Then I think I skipped the statement C. Triglycerides may have double bonds. That is correct, ne? So we have A, C, D as the answer. Spider silk is people secondary structure. Secondary structure. Now they say bending and the folding goes for the tertiary structure, ne? That goes for the tertiary structure. So we'll take that as wrong. Question number 15. Which of the following features are commonly Euglena, Paramecium and amoeba? Following features are common to Euglena, Paramecium and amoeba. Freshwater species present unicellular. Freshwater species present and unicellular. Correct, Ned? And then no cell wall pellicle present. No cell wall pellicle present. Amoeba don't have a pellicle. Contractile vacuole present and heterotrophic. Now Euglena is mixotrophic. Freshwater species present, nuclei present. Freshwater species present. Nuclei present. Nuclei. Euglena has a nucleus, one single nucleus. Paramecium has nuclei, two nuclei. All are parasite. Wrong. So what's your answer? I think I can pick first one as the best answer. Do you agree with me? Right, we'll check people. The structurally answers, the marking scheme is given. The marking is given. Now, what are your difficulties, people, in doing this paper? What are your difficulties? Time management, what are your issues, people, at present? I think I should ask this from the people who have not actually answered and submitted the papers. Maybe some of you have not answered the paper, and then what is your the excuse? What is your problem, or maybe what is your issue, people? Can you let me know? What sort of problems you have? Too much. We can't study and finish within the week. So you are giving 2 sections, so it's too difficult to study 2 sections per week. This is once in 2 weeks, you have to study 2 sections. Huh? And nothing, just to learn by heart. What are your problems? Well, let me know. So I can adjust, I can adjust this work as the way you want. If you don't have physically time to come and write this, we can do it now online. We created folders so that there was an email issue. Right now we sort that out also. So we have to adjust our time. So I can come and adjust your time also if you like. So I did the paper, but I had some issues in remembering the important points. I didn't find any problem in attempting the paper. So will you upload the marked papers? I have uploaded the marked papers. Some of you have received it. Please check in the folder, Dineth. Can you, sir, cannot we do this physically? Yeah, we can do this physically also. You have to come on Friday. Come on Friday afternoon, morning, whatever the time. We can let you come and write this and go. Right? So doing online is embarrassing. That is different people. Or anything as the way you like. We can hand over papers to you, sir. Can we hand over? Yeah, no problem. Right? No problem. I can look into that also. But then thing is, people, when different people are doing it different ways, then it takes time. You hand over it to me, we have to mark it and then hand over it back to you. You are not coming on that day. I forgot to bring the papers on a particular day, right? And then that's going to simply drag, so we won't be able to finish it, marking and then discussing it comfortably like this. So we can come on Friday and come anytime and write it. How? Yeah, it doesn't matter. You come on Friday because the 2025 people are writing it on Friday, their papers. So I can just send this paper also, but then once in 2 weeks you have to come and write. No problem, right? So we are handing over the paper on Saturday, right? You can bring the paper here and then come and answer and then hand it over, right? Okay. Next one, people. State main properties of water related to each of the function given below. Each of the function given below. First one is ability to transport water as continuous column through the xylem against the gravity. What is that, people? Cohesive behavior. I'll go with the scheme now. Right, cohesive behavior. Cohesive behavior. Second one, water will function as a thermal buffer in living systems. Ability to moderate temperature. Then indicate the function performed by polymeric molecules of indicate the function of the function performed by polymeric molecule fructose, storage material in daily actives. State the monomers of the following polysaccharide: inulin, fructose, pectin, galactonic acid. Write structural polysaccharide in animals. Write structural polysaccharide in animals. Chitin. What is the building unit in above mentioned polysaccharide? Glucosamine. Name the storage carbohydrate of milk, lactose. Name the monomer of polysaccharide which is component of the fungal cell wall, glucosamine. Now they ask the monomer. This material is chitin, so the monomer is glucosamine. Monomer is glucosamine. Right, then people, what is the main structural difference between saturated fatty acid and unsaturated? So we learned that people in the hydrocarbon tail, when double bonds present, it is unsaturated, right? So unsaturated fatty acids have double bonds in their hydrocarbon chain. So you obtain 2 marks for that. You learned the right features that distinguishes phospholipid from fat. Feature that distinguishes phospholipid from fat. Fat consists of glycerol and fatty acids, and phospholipid is composed of 2 fatty acids and 1 phosphate attached to 1 glycerol. Phospholipids and fat. Now you know, in fat molecules, 3 fatty acids present, and phospholipids you can see that only 2 fatty acids and another is a phosphate group. Write down the reason why 2 ends of the phospholipid behaves differently. You know, people, the carboxyl group is polar because of oxygen, and then the hydrocarbon tails are non-polar, right? The hydrocarbon tails are hydrophobic, while phosphate group and the attachment are phospholipid. Okay, let me take this once again. Write down the reason for 2 ends of phospholipids, so that the phosphate group and the attachment, the choline part is polar and hydrophilic, rest of the part is non-polar. So what do you have now, people? Amphipathic molecule. Polar, non-polar, both features are there. Name 3 major types of lipids found in organisms. Now you have to write fat, phospholipids and steroids. Right, and then part B, what is meant by denaturation of proteins? Denaturation of proteins. Protein denaturation. What is that, people? The loss of specific chemical three-dimensional shape due to alternation of weak chemical bonds and interactions. Now you have to write, when you write the answers, people, you have to write it is as it is given in the resource book. At least you have to use the keywords and then write. Okay, you can't simply ignore all these things and then write something that you memorize. So try to answer repeatedly, write them, study them repeatedly, and try to memorize these phrases and all, people. Next one, state the differences between hydrogen bond from bonds form the secondary and the tertiary structure. What do you say, people? In the secondary structure, hydrogen bonds are formed between the distal amino acids of the same polypeptide chain. And in tertiary structure, people, the hydrogen bonds are going to form between the R groups. In tertiary structure, hydrogen bonds are formed between R group amino acid. Right, question B, give a protein that other than hemoglobin with quaternary structure, that is collagen. Collagen. State 3 agents affecting the denaturation of protein: high temperature, high energy radiation, strong acids, alkaline, high concentration of salt, heavy metal, organic solvents, detergents. Any 3, you get 3 marks. Name a protein that has defensive role in man. What do you take, people? Immunoglobulins. State the protein identifying test, BioEve test. How do you perform BioEve test? You take protein solution, diluted protein solution, add few drops of copper sulfate and the sodium hydroxide, then heat it, and then you can see that this color change occur from blue to purple. So that's how it happened. Then moving to next section, part C, how condensation occurs between nucleotides. How condensation occurs between nucleotides. You know, people, OH group attached to the so can't we say leukocytes for the protein with defensive role? No. Leukocytes are white blood cells. White blood cells are not a protein, right? White blood cells are not a protein, it's a cell, right? So you can't write that. How condensation occur between nucleotides. OH group of the phosphate of one phosphonucleotide and OH attached to the third carbon forms the phosphodiester bond. Right. Next one, people draw the DNA molecule, double helix, and label it. So, expecting people some details here, right? You have to draw the DNA molecule. Even if you draw it like this, it is okay, right? Like this. But you have to mark this 3' end and the 5' end. And then you have to label people sugar phosphate backbone like that. Then you have to show people this the base pairing, right? Adenine, thymine, guanine, cytosine. Show the hydrogen bonds between them two hydrogen bonds, 3 hydrogen bonds there. And then that's all. You don't have to like draw a diagram which is similar to the resource book one. No need. But then certain things you have to label. Then you can label a base pair. The guanine, cytosine, adenine, thymine, they are base pairs. So here we gave 2 marks for that. What arrangement is said to be anti-parallel? Two sugar phosphate backbones run opposite direction from each other. That we call as anti-parallel. State 3 differences between DNA and RNA. DNA double-stranded, RNA single-stranded. DNA consists of adenine, guanine, cytosine, thymine, and no uracil. Instead, RNA has adenine, guanine, cytosine, and uracil, and no thymine. Sugar molecules in RNA is ribose, while DNA is deoxyribose. State 3 nucleotides other than those found in nucleic acids, NAD plus, NADP, FAD. So that's the end of the first question. That's the end of the first question. Right. Then we move to people second question. Second structured question. State major function of each of the following structures. Major function of each of the following structures. Oral groove, ingestion of food, pseudopodia, locomotion and feed. Right, 2 key characteristics of kingdom Protista. Most of them are unicellular, although there are some colonial and multicellular species. Most are unicellular. It is a polyphyletic group found in fresh water. Unicellular, colonial or multicellular. Some are phototroph, heterotrophic and mixotrophic types are there. What is meant by polyphyletic? That means the organisms have originated from more than one ancestor. That you call as a polyphyletic. And then state 1 special structural feature present in each of the following. Euglena, they have a pocket at one end, and then 1 or 2 flagella arise from that. Anything else you have written, people? Special structural features, maybe you can say chloroplast. Chloroplast available. Paramecium, 2 types of nuclei, meganucleus and micronucleus. And then pseudopodia, which are used to locomote and feed. Then what is meant by mixotrophs? What is meant by mixotrophs? Combination of photoautotrophic and heterotrophic nutrition is called as the mixotroph. State how the locomotory structures of Euglena, Paramecium and amoeba differs from one another. Differs from one another. Locomotory structures, Euglena, they have flagella. Paramecium has cilia. Amoeba use pseudopodia. So this is what you have to write. So for euglena, can we write eye spots? Yeah, fine. Eye spots you can write. It's mentioned in the resource books, you can write. No problem. State organism belongs to protist, which have following features. Only contractile vacuole present. Euglena. Both marine and freshwater members are present. Amoeba, diatom. Meganucleus present, paramecium. Possess no definite shape, amoeba. Multicellular. Ulva, gelidium and sargassum. So I have marked the people and the total marks that you are receiving for answering that. I think in your mark papers the total marks are mentioned. If not, tell me. Right, we'll fix it in future. Complete the following table. Ulva cell wall component is cellulose. Gelidium also cellulose. Sargassum, cellulose and alginate. Diatom, pectin and silica. Then storage food, ulva, starch. Gelidium, Floridian starch. Sargassum, laminarin. Diatom, oil and chrysolaminarin. Photocytic pigments, chlorophyll A and B. Chlorophyll A and D for gelidium. A and C for sargassum. Diatom A and C. Right, next one, state photosynthetic pigments that are present in both sargassum and diatom. Chlorophyll A, C, and xanthophyll. Sargassum and diatom. Sargassum and diatom. You can't take carotenoid, ne? Carotenoid you cannot take, right? So chlorophyll A, C, xanthophyll. Right, next one, complete the following dichotomous key. Complete the following dichotomous key using appropriate numbers. Multicellular body. I think people you can remember the way to deal with this the dichotomous key. How many of you got this answer correct? Dichotomous key question. How many of you got this correct? Vinuk got it correct. Amira got it correct. Very good. Right. Okay. So here they say that multicellular body. Who has multicellular body? Sargassum is multicellular, multi. Amoeba not, diatom not, gelidium multi, ulva multi, euglena not. So multicellular body we take to number 2. Then multicellular body absent we take for number 4. Then stipe present, sargassum ne. Stipe absent number 3. So sargassum, then gelidium and ulva, no stipe. Then phycobilly protein present in gelidium. Ulva phycobilly protein absent. Then autotrophic organisms out of this amoeba, diatom and euglena. Autotrophic organisms number 5. Why? Diatom and euglena both are autotrophic. Euglena has ability. And then autotrophic, not autotrophic, amoeba. Then cell wall present diatom, euglena, cell wall absent. So I think you can do that easily. They are not complicated questions. But then people, you have to have some proper plan to work and get ready. It's like 2 sections per 2 weeks. People is a very elementary task. Elementary neither. So the nursery work. So I didn't receive my marked paper. Check in the folder. Hafik, check in that folder. Did you check? Did you check in the folder? Is your paper is there? Is your unmarked paper is there? Yes, I'll check then the issue. I'll check the issue, okay? And right, then we will, so next week from what topics are we going to get the questions? Let me tell you. Paper 6. Paper 6. Paper 6. Paper 6. Paper 6. Shall I send that to the group? I'll send it to the group, okay? To check that paper. I'll send it to the group, okay? Paper 6. Paper 6. Paper 6 from enzyme, from enzyme lesson, and I think classification. Yeah, classification. Classification part. Okay. So can I get this week month PDF? What is that? This week month PDF means what? Right. Let's talk about that, people. Briefly explain the structure of structural details of lipids. Structural details of lipids. Diverse group of hydrophobic molecules, large biological molecules, but not polymers. Right. Not polymers or macro. PDF, all PDF for online students I have uploaded in the website. You can get them from the website, okay? Consist of carbon, hydrogen, oxygen. Hydrogen oxygen ratio is not 2:1. Comparatively more hydrogen than oxygen. So briefly explain the structural details of lipids. Structural, neither. Structural details only. Right? Structural details only. Biological important types of lipids are fat, phospholipids, and steroids. Fats are made up of glycerol and fatty acid. Glycerol belongs to alcohol group, having 3 carbon, and then having single hydroxyl group. Fatty acids are hydrocarbon chains with a long, usually 16 to 18 carbon skeleton with carboxyl group attached to one terminal. Fatty acid molecule bind to each hydroxyl group, glycerol, by ester bonds. The resulting fat molecules are called as triacyl glycerol, or else you can say triglyceride. And then hydrocarbon chains of fatty acid contribute for hydrophobic nature. Based on the nature of the hydrocarbon, we can classify people as saturated fat and unsaturated fat. Saturated fat, the hydrocarbon, the fatty acid of the hydrocarbon tails of fatty acids are containing, not containing any double bonds. And unsaturated means they have double bonds. Examples for unsaturated ones, you can say butter. Unsaturated ones you can take as vegetable oil. Then unsaturated fats are classified based on the nature of the double bond, cis ones and the trans ones present. Cis and trans fat. Phospholipid is composed of two fatty acids, one phosphate group attached to one glycerol molecule. Right, and then choline. Briefly explain the functional importance of proteins. Briefly explain the functional importance of proteins. Functional importance of proteins. Some are catalysts, enzymes. Some are structural keratin. Some provide strength and support. Storage, albumin. Then milk storage, casein. Transport, hemoglobin. Transport, serum albumin. Hormone, insulin glucagon. Contractile, actin myosin. Defensive, immunoglobulin. Right, then people, the second question, explain the diversity of algae. Vulva, gelidium, sargassum, diatom. So what you have to write here, the diversity of algae, okay? So you know they are diverse in the cell wall material, storage material, pigment molecules, unicellular, multicellularity, living environment. So they are diverse. So explain the diversity of algae. Vulva, macroscopic and macroscopic. Cell wall present, made up of cellulose. Blade-like thallus, root-like holdfast, storage is starch, chlorophyll AB, green color. Gelidium, marine, mainly cellulose wall. Multicellular thallus, holdfast present. Thoridian starch as storage. Photosynthetic pigments and chlorophylls, phycobilly proteins present. Greenish red color, sargassum, marine, cell wall made up of cellulose and alginic acid. Comparatively large and complex, multicellular thallus, root-like holdfast, stem-like stipe, leaf-like blade, storage is laminarin. You know, sargassum has stipe, ne? And then what do you call, floats are present. And then photosynthetic pigments, chlorophyll, AB, xanthophyll, appear in olive green color. Then diatoms, aquatic, freshwater, unicellular. And then cell wall has silica, pectin and silica, glass-like cell walls. And then highly diverse group, markings on the cell, oil and chrysolaminarin, chlorophyll, AC, carotene, xanthophyll, golden brown in color. Golden brown color. Right, those are the things that you need to know. Right, so we answered this paper. So get ready for the next paper. Get ready for the next paper. Don't give excuses. Somehow people, you have to get used to this answer, right answer, and have people some well planned. You have to have a timetable, then proper working schedule you have to have. That takes you to the success. If you think people, the person who's not having any plan, not working properly, sleeping too much, oversleeping, and people enjoying the life, then they can't get through. Right? So the only way that is available is to study, practice, and the hard way. So I'll see you all on weekend class, right? As usual on Saturday. So I'll give you the paper number 6 on Saturday, right? Okay, then thank you very much. I go on. Bye-bye.